Okay, today I want to talk about a urinary system. First, you need to know that in our urinary system, we have two kidneys, right and left kidney, which are located on both sides of our abdominal cavity, out of our peritoneum, which we call them retroperitoneal organ. From the kidneys, we have two tubes, which we call them ureter. The ureter come down to a sac, which is urinary bladder. The urinary bladder can store urine for us. And after that, we have a small tube, which we call it urethra, which can help the urine go out of our body. In this part, I want to talk about the anatomy of each of these parts for you. First, you need to know that the scientific study of the anatomy and physiology and also pathology of our kidneys is called nephrology. And the urology is the branch of medicine which works with the male and female urinary system and the male reproductive system. Our urinary system has different parts. It contains two kidneys, ureters, bladder, and urethra. And you know that the function of our kidney is filtration of the blood. Most of the water and solutes which is filtered in our kidney return to our blood by a process which we call it reabsorption. And I will talk about it next week for you. And our kidney can do homeostasis of the fluid which we have in our body. It can manage the homeostasis for the fluid, electrolyte, pH of our blood, and the volume of water. And I will talk about them in the physiology of the kidney. In this picture, you can see the organ which we have in our urinary system. Here you can see left, and here you can see the right kidneys. You have these two tubes which come down from the kidney, and they are ureter. After that, they enter this sac which we call it urinary bladder, and after that we have urethra. These are the different parts which we have in our urinary system. In this slide, you can see the function of our kidney. First, your kidney can do regulation of the ions which we have in our blood. The amount of sodium ion, potassium, chloride, calcium, and phosphate. The other function of our kidney is regulation of the blood pH. When the pH of our blood becomes acidic, our kidney can secrete more hydrogen ion to our urine, and it can conserve more carbonate. And when our blood become alkaline, then it can conserve hydrogen ion and it can secrete more carbonate to the urine. The other function of our blood is the regulation of our blood volume. When you drink more than 5-6 cups of water during the day, your urination increases. And when you are dehydrated, the amount of your urine decreases. This is the meaning of regulation of your blood volume. Because in your body, you need about 5 liters of blood. And if the amount of blood increase or decrease, this is the function of our kidney to do homeostasis. The other function of our kidney is regulation of our blood pH, and it's dependent on the blood volume. When the volume of the blood increase, the blood pressure increase, and when the volume of the blood decrease, the blood pressure decrease. So by adjusting the volume of the blood, your body can regulate the pressure of the blood too. It can maintain the blood osmolarity by a regulation of a loss of water and solutes. It can produce different hormones. You know that calcitriol is the active form of vitamin D, which become active in your kidney, and vitamin D is very important for absorption of calcium ion. Without vitamin D, you cannot reabsorb any calcium ion. The other hormone which is secreted from our kidney is called erythropoietin. And you know that the erythropoietin is the hormone which can go to your bone marrow and stimulate the pluripotential stem cell to change to myeloid stem cell, and the myeloid stem cell go to the process of erythropoiesis and make red blood cells for us. The other function of our kidney is regulation of blood glucose, because our kidney can do gluconeogenesis. It can make glucose, so it can regulate the blood sugar, and excretion of metabolic waste and foreign substances like drugs and toxins are the other function of the kidney, and I will talk about this filtration and excretion next time in the physiology of kidney for you. The kidneys are a retroperitoneal organ. You know what is the meaning of retroperitoneal? It means that they are located out of peritoneal cavity. Digestive system, when I talked about the peritoneal membrane, I told you that some organs are located out of this peritoneal membrane, and we call them retroperitoneal, and our kidneys are examples of these organs. They are protected by our lower ribs. Rib number 11 and 12 can protect our kidney. The kidneys are placed between thoracic vertebrae number 12 to lumbar vertebrae number 3, and the right kidney is placed lower than the left kidney because in the right side of our body, right side of our abdominal cavity, we have liver which can make pressure to the right kidney and help it to go down. In this picture, you can see the location of right and left kidney. You see that the right kidney is a little lower than the left because in this part we have liver which make pressure on the kidneys. In the medial side of your kidney, you can see the indented part. These indented parts are called hilum or hilus. And this part is the part where the blood vessels can come inside or go out of our kidney, and the ureter can leave the kidney. You need to know that your kidneys are covered by fat pad, and this fat pad can do protection of the kidney and make a cushion around our kidney. And the size of our kidneys are 12 by 6 by 3 centimeter, and the shape of our kidney is bean shape or kidney shape. In this picture, you can see the anatomy of the kidney from outside. You see that it's bean shape. In the medial border, it has the indentation, and this indented area is called hilum of the kidney. You see that the renal artery come inside the kidney, and the renal vein leave the kidney. And here you can see the ureter which leave the kidney. Also, the nerves and lymphatics pass through this hilum, and they can do innervation of the kidney, and the lymphatics can take the lymph out of the kidney. In this picture, you can see the layers around the kidney. First, you need to know that your kidney is covered by tissue capsule. The tissue capsule of the kidney is the fibrous connective tissue. This is dense, irregular connective tissue, and this connective tissue can maintain the shape of the kidney, and it can protect our kidney. After that, we have some fat or adipose tissue around the kidney, which we call it adipose capsule. The other name of this adipose capsule is perirenal fat. This is a thick adipose tissue which covers the kidney from outside, and it can do suppression and protection of the kidney. And after that, we have a fascia or renal fascia around the kidney. This is the other connective tissue which can protect the kidney and also attach the kidney to the neighboring muscles, and it can fix the kidney in its own place. These are the supportive structures which we have around the kidney. And from superficial to deep These connective tissues are renal fascia, which can anchor the kidney to other structures, adipose capsule, which can do protection and anchoring, and the renal capsule, which continues with the ureter, and it can provide shape of the kidney for us. Now, I want to talk about the internal anatomy of the kidney. Look at this picture. In this picture, we have a frontal coronal section of the kidney. You know that the frontal section can divide the kidney to anterior and posterior portion. When we do this cut, you can see two major parts in the kidney. One part is located outside, we call it renal cortex, and one part is located inside, and we call it renal medulla. So inside the kidney, we have two parts. Cortex is located outside, and the medulla, which is located inside. In the medulla, if you pay attention, you have two colors. One part of medulla is pyramid shape, triangle shape in this picture, and it's darker. One part of medulla is lighter, and the color is very similar to the color of cortex. So we can divide our medulla to two parts. One part, which is darker, is called renal pyramid, and one part, which is lighter in color, is called a renal column. So this is the first division which we have for our kidney. First, you have a tissue capsule, a fibrous connective tissue around your kidney. Then you have renal cortex, and after that you have a renal medulla, and in medulla you have renal pyramid and renal column. The other thing which is important to know is this. The functional unit of our kidney is a structure which is yellow in this picture, and we call it nephron. In each of your kidneys, you have about 1 million nephrons, and these nephrons are responsible for making urine for you. I will talk about this structure later for you, but you see that each nephron is open to the tip of your renal pyramid. And we call this tip of renal pyramid renal papilla. And I will show this part to you in the next slides. Again, in this picture, you can see the cortex, you can see the medulla. In the medulla, you have renal pyramid and renal column. And I told you that the tip of each pyramid is called a renal papilla, as you see in this picture. This renal papilla open to a duct, a small duct, which we call it minor calyx. After that, these small minor calyx open to the larger duct, which we call it major calyx. All major calyx open to a sac-like structure, which is renal pelvis, and the renal pelvis is continue with the ureter, and the ureter can bring the urine to the urinary bladder. So what happened here? This is the nephron. As you see in this picture, some parts of your nephron is located in the cortex, and some part of the nephron is located in the renal pyramid. After that, the end of your nephron, which open to the minor calyx, can make the renal papilla, a small hole here. The renal papilla, I will talk about collecting duct and papillary duct later for you. This renal papilla open to minor calyx. Minor calyx combine together and make major calyx. Major calyx can make renal pelvis, and renal pelvis can open to the ureter, and ureter merge to urinary bladder. These are the different parts which we have in our kidney. This is the real kidney. In the real kidney, again, you can see the cortex, which is located outside and it's lighter in its color. The cortex has granular appearance. And I will tell you why do we have this appearance in this part. After that, you can see the medulla. In the medulla, you have renal pyramid, and between this pyramid, you have renal column. The renal column has the other name, which we call it renal column of Bertin. And the color of this part is the same as cortex. Here you can see the renal pelvis, and the renal pelvis continue with the ureter. This indented part is called hilum of the kidney. This is the fixed kidney. If we had face-to-face class, we open the kidney and see the different parts of the kidney. Here you can see the cortex, and this part is called medulla. You can see the minor calyx and also major, and this is the renal pelvis. I will post one video for dissection of the kidney later today for you. This is the other picture of the kidney. I repeat all of these structures for you because it's very important for your lecture and also your lab. You can see the capsule. You have the cortex of your kidney. Here you can see the medulla. In the medulla, we have renal pyramid and the renal column. The tip of each renal pyramid is called renal papilla. The renal papilla open to these small ducts, which we call them minor calyx. The minor calyx open to the major calyx, and these major ones are open to the renal pelvis, and the renal pelvis continue with the ureter. These are the different parts which you need to know up to now. Yes, Ola, what is your question? You can turn on your microphone. No questions? Okay. These are the internal anatomy of the kidney. And between these ducts, as you see here, we have adipose tissue. These parts with adipose tissue are called a renal sinus. And I think I have one slide to show you the renal sinus. The cortex and the medulla of the kidney can make renal parenchyma. Parenchyma are the functional part of our kidney, the part which can make urine. I talked about parenchyma and stroma before for you in the lymphatic system. I told you that the stroma is the connective tissue, the capsule, and the invagination of the capsule inside, which can make the framework and the shape of the organ for you. And the parenchyma is a part which is functional part. And the parenchyma of our kidney contains about 1 million nephron. And nephron is the structure which can make urine for us. This is the other slide which shows you the minor calyx. In each of our kidneys, we have about 8 to 18 minor calyx. They are different in individuals. We have 2 to 3 major calyces, and these major ones can combine together and they can make renal pelvis, and renal pelvis continue with the ureter. Yes, Ola, do you have any question? You need to turn on your microphone.
I'm sorry. I'm sorry. It was a mistake.
Okay. And as I told you before, the renal sinus are these spaces which we have between these ducts, and it's full of adipose tissue. I'm going to talk about the blood and nerve supply to our kidney. Your kidneys are less than 1% of your total body mass, but they can receive 20 to 25% of your cardiac output. It's a large volume of blood for these small organs. We need to have many blood vessels inside the kidney to bring the blood here because we need to filter the blood in our kidneys. And also we have nerve supply for our kidney, and the renal nerves can carry sympathetic outflow to our kidney, and they can regulate the blood flow to our kidneys, and I will talk about it later for you. Now I want to talk about the blood vessels of the kidney. This part is very important for your lab and also your lecture. And you need to know the name of all of these blood vessels in order in your kidneys. First, you need to know that the renal artery is the blood vessel which brings the blood to your kidney. When this renal artery comes to the kidney from the hilus, it divides into some branches. These divisions are called segmental arteries. The segmental arteries go to the renal columns between the renal pyramids. And when they move in this renal column, we call them interlobar arteries. The interlobar arteries come up, and between the medulla and cortex, they make the arch-shaped structure like this. We call it arcuate arteries. And from these arcuate arteries, you can see some branches which come to the cortex. These branches are called interlobular arteries. The interlobular arteries have the other name, which we call them cortical radial arteries, and you have it in the other slides. Now, I want to talk about these cortical radial arteries. Look at this picture. The cortical radial arteries can make a branch which we call it afferent arteriole. This afferent arteriole can come to a space in our nephron and make a network of capillaries. The network of capillary in our nephron is called glomerulus. And from the glomerulus, the other artery come out, which we call it efferent arteriole. This efferent arteriole starts to wrap around the nephron. If this wrapping takes place in the cortex, we call it peritubular capillary. If it happens in the medulla, we call it vasa recta. And after that, these peritubular capillary and vasa recta can make the veins. So, what happened here? I want to repeat it for you. The renal artery comes to our kidney from the hilum. It makes some segments, some branches, which we call them segmental arteries. These segmental arteries come in the renal column between the renal pyramids, and it can make interlobar arteries. Interlobar arteries in the place between the medulla and cortex can make the arch-shaped structure, and we call it arcuate artery. And the arcuate artery makes some branches here, which are interlobular arteries. Now, the next part is here. Interlobular arteries can make afferent arteriole. Afferent arteriole makes glomerulus inside the nephron. The glomerulus makes the efferent arteriole. and the efferent arteriole start to wrap around the nephron. The wrapping in the cortex is called peritubular capillary. In the medulla, we call it vasa recta. And after that, the peritubular capillaries and vasa recta can make the veins. The veins are here. First, in the cortex, you can see these small veins. We call them interlobular veins, or the other name of these interlobular veins are cortical radial veins. These cortical radial veins can make the arch shape structure near and adjacent to arcuate arteries, and we call them arcuate veins. After that, the arcuate veins can make interlobar vein adjacent to interlobar arteries. Then we have, in this picture, it shows you segmental veins, but really we don't have any segmental veins. And interlobar veins, all of them can combine together and make a renal vein which leave the kidney. Some authors agree that between interlobar and renal vein we have segmental, but the others don't agree this part, and they told that these interlobars can join together and make the renal vein which leave our kidneys. So you see that we have many blood vessels in our kidneys, so they can carry about 20 to 25% of our resting cardiac output in each time, and they want to filter our blood during this time. Now, I want to talk about the structure of the nephron. Before I talk about the structure in this picture, I want to show you first this picture, and then I come back to the previous one. This is the cartoon of the nephron, and I told you that the nephron is the functional unit, and in each of our kidneys we have more than 1 million nephrons. The nephron has different parts. The first part of the nephron is this structure. This structure of the nephron, which you can see here, is called renal corpuscle. The renal corpuscle has two main parts. One part is this beige part here. We call it Bowman's capsule or renal capsule. The other part is this capillary network inside, which we call it glomerulus. So, glomerulus and the Bowman's capsule together can make renal corpuscle. And this is the renal corpuscle. As you see in this picture, the renal corpuscle is circular in its structure. Because of this structure and because this part is located in the cortex, you can see granular structure in the cortex of the kidney. After this Bowman's capsule, you have a duct here, which we call it proximal tubule. The proximal tubule has two parts. One part is located in the beginning of this tubule. We call it proximal convoluted tubule because it's coiled-like structure. And after that, this tube becomes straight, and we call it the proximal straight tubule. After that, we have a loop-like structure. This loop structure has one descending limb which come down, loop of Henle which is U-shape, and the ascending limb which goes up. This ascending limb has one thin part, one thick part. And after that, we have distal tubule. This distal tubule again has two parts. One part is straight. It comes, we call it distal straight tubule. After that, we have distal convoluted tubule, and our distal convoluted tubules open to a collecting tubule. Distal convoluted tubule of about 5 nephrons combine together and make one collecting tubule. Now, I go back to that slide, this one. This is the structure of your nephron. As you see in this picture, some parts of your nephron is located in the cortex. Here, let me mark it for you. This part is the cortex of your kidney, and some parts of your nephron are located in the cortex. And the next part is this part. This part is the medulla of your kidney. And you see that some parts of your nephron is located in the medulla. Now, look at here. In the cortex, you have a renal corpuscle, this round structure. The renal corpuscle has one cup-shaped structure here. We call it Bowman's capsule. And in this capsule, you have capillary network, which is glomerulus. Glomerulus and Bowman's capsule together are called a renal corpuscle. Then we have proximal convoluted tubules, coiled. You see it. Then we have proximal straight, which comes down. Then you see that your nephron enter the medulla. First, we have descending limb. After that, you have loop of Henle, and then you have ascending limb, which comes up again to the cortex. Then you can see the distal straight. Then we have distal convoluted, and after that we have collecting duct. See that the distal convoluted tubule, all of them, let me mark them for you. This is 1, this is 2, this is 3, and this one is 4. Up to 5 distal convoluted tubules combine and make one collecting duct or collecting tubule. These collecting tubules from different nephrons and different parts combined together and make papillary duct in each pyramid. And these papillary duct open to the renal papilla in the tip of each pyramid, and as you see here, this part open to the minor calyx. And then minor calyx make major makes a renal pelvis, and the renal pelvis can make ureter. And the urine can go to the urinary bladder, store there, and when the amount of urine reach its volume, which can make pressure on the wall of bladder, then we have the process of micturition reflex or urination. So in this picture, you see that some parts of your nephron are in the cortex, some parts of your nephron are in the medulla. Let me finish this part and then I answer your questions. Then you see that in this structure we have the renal corpuscle here. The renal corpuscle is composed of the renal capsule or Bowman's capsule, and also we have this glomerulus. Then you can see the proximal convoluted tubule, proximal straight tubule, descending limb, loop of Henle, ascending limb, then distal straight, distal convoluted, and then you can see the collecting duct. The collecting ducts merge together and make papillary duct. And these papillary duct open to the renal papilla, and the renal papilla can open to the minor calyx. These are the different parts of the nephron, the functional unit of your kidneys, which can make urine for you. In this picture, I want to show you some blood vessels too. Do you remember I told you that between cortex and medulla, we have the artery, which we call it arcuate artery. This arcuate artery can make the interlobular artery or cortical radiate artery. This cortical radiate makes afferent arteriole. Afferent arteriole makes glomerulus, and then efferent arteriole come out of renal corpuscle. This efferent arteriole start to wrap around the nephron as you see here. The parts which you have in the cortex is called peritubular capillary. The parts which you have in the medulla are called vasa recta. And as you see here, all of these peritubular capillary and vasa recta combine and make this vein. This is interlobular vein or cortical radiate vein in the cortex. It makes this arcuate vein, and then the arcuate vein can make the interlobar veins. Professor Imholtz talks about all of these structures in the models of the lab which I posted for you before. In our kidneys, we have three major processes. One process is called filtration. And as you see in this picture, this is the renal corpuscle. Here you have Bowman's capsule, and this is the glomerulus. The filtration, the first physiological process happens here in the renal corpuscle. The blood is filtered to Bowman's capsule. But most part of the blood which come to Bowman's capsule are essential for our body. So we have the other process which we call it tubular reabsorption. The material which we need for our body reabsorbed from this tubule to the blood vessels again. And after that, we have the other process which we call it tubular secretion. It means that still we have some waste products like urea, uric acid, creatinine, some toxins in our blood which need to go to our nephron and excrete from our body. So we have the other process which we call it tubular secretion, and by this process these waste products can go to the nephron again. So this is why do we have many blood vessels around the nephron. And the renal nerves are originate from the renal ganglia. They can make a renal plexus, and this renal plexus can enter our kidney from the renal hilus, and the vasomotor nerves can regulate the blood flow, the blood pressure in our kidney. Sometimes we need more filtration, sometimes we need less filtration, and these nerves can regulate the diameter of our blood cells and the pressure in the kidney. So we have different of filtration in our nephron. I will talk about it a little for you. I talked about this structure for you. The cortex kidney, you have some parts of your nephron are in the medulla. Most part the filtration take place in our renal corpuscle, and all of these tubes help reabsorption and secretion, which you learn about it later. Now I want to talk about this picture for you. This is renal corpuscle. The renal corpuscle has the glomerulus. This is the capillary which we have here. This is the afferent artery which bring blood. This arteriole makes capillary, and after that we have the other arteriole that leaves part. You know that. For the capillary, we need one artery in one side and one vein on the other side. Arteriole makes capillary, and let me draw it here for you. We need to have one arteriole here. This arteriole should make a capillary network in the center for us, and after that one vein or venule should leave this part. But in this structure you see that we have capillary network in between, but on both sides we have arteriole. This is a specialized pathway of the blood vessels which we have in our nephron, and we call it admirable system. On both sides of capillary we have arteriole. This is one point for this picture. The other point is about this Bowman's capsule. The Bowman's capsule, as you see in this picture, has two layers. One layer attached to the glomerulus, we call it visceral layer, and the cells which can make a visceral layer, as you see here, are simple squamous cells, simple squamous epithelium, and these epithelial cells contain many feed processes. The feed process of these cells can change the name of this visceral layer to podocyte. Podocyte means the cell with many feet. I will show you in the next picture. After that, you can see this space of Bowman's capsule, and then again you have the other layer, which is parietal layer. This parietal layer of Bowman's capsule is simple squamous epithelium again. So both layers of your Bowman's capsule are covered by simple squamous epithelium, visceral layer, or podocyte, and this is the parietal layer. And you see that immediately after our Bowman's capsule is finished and the proximal tubules start, the epithelium change. The epithelium change to simple cuboidal epithelium. Here we have simple squamous, so filtration of the blood takes place easily. And in the tubules, like proximal, loop of Henle, and distal, we have the process of reabsorption. So we need simple cuboidal epithelium in this part. In this picture, it shows you the pathway. In the normal pathway is the arteriole in one side, capillary in the middle, and the venule on the other side. But in the renal corpuscle, we have afferent arteriole. It makes capillary, and efferent arteriole leave. This system is called admirable system. I talked about it before in the blood vessels pathway for you. I will talk about these slides on the pictures later. You need to know that we have two types of nephron in our kidneys. One type of nephron is called cortical, and the other type is called juxtamedullary nephron. Now I want to show you their differences. Look at these two pictures. The first picture shows you the cortical nephron, and the next one is juxtamedullary. The cortical nephron is shorter than the juxtamedullary nephron. The cortical nephron is located in upper part of your cortex, and the juxtamedullary come down near medulla. Juxta means near, and juxtamedullary are the nephrons which are very near to the medulla. Now, what is the difference between them? First, you need to know that your cortical nephron is about 80 to 85% of your nephron. They can make the standard urine in the volume and in its color. But the juxtamedullary nephron, which are about 15 to 20% of your nephron, can make concentrated urine. By this long loop of Henle, they can reabsorb as much as water they can. So the urine which is produced at the end is concentrated. So when you are fasting, when you have a dehydration, when your blood pressure change, at this time your juxtamedullary nephron start to work and make concentrated urine for you. And when you're normal, drink plenty of water during the day, your cortical nephron works and they can make a standard urine for you. And in the next slide, it talks about the cortical nephron, which are about 80 to 85%, and they create urine with osmolarity, which is very similar to to our blood, and they have short loop of Henle, which are located in the upper part of our medulla. And the other one is juxtamedullary nephron. They are located in the deep cortex. They have long nephron loop. They can receive blood from peritubular capillaries and vasa recta, so they can do many reabsorption and they can make concentrated urine. And you should know that the animals like camels who live in deserts have more juxtamedullary nephron because they don't have access to water all the times. So they need to preserve water as much as they can in their body. So their nephron, most of their nephron are juxtamedullary nephron. This is, in this picture, you can see the histology of renal corpuscle. In this histology, you have the afferent arteriole, which bring the blood inside the glomerulus. This is your glomerulus. And this one is the efferent arteriole, which leave your renal corpuscle. This is your Bowman's capsule. And as I told you, our Bowman's capsule has two layers. One layer of the cells are simple squamous cells with many feet process, as in this picture. We call them podocyte. And this parietal layer is simple squamous epithelium, and both of them together can make this Bowman's capsule for us. Here you can see the podocyte cell, the cells with many feet process, and by this feet process they can hug and attach to the glomerulus. Between these cells we have some small cells like this one or this one, which we call them mesangial cells. The mesangial cells have actin and myosin inside, and you know that when your cells contain actin and myosin, it means that they can do contraction and relaxation. By this contraction and relaxation, they can do contraction and relaxation of the blood vessels, so they can change the blood pressure inside of this glomerulus, and they can change the amount of filtration for us. The other thing which is important to know is this. The glomerular capillaries are fenestrated capillaries. You know that the fenestrated capillaries are the capillaries which contain pores or fenestration in the endothelial cells, and they have a complete basement membrane. Some particles which are smaller than these pores can easily pass through this fenestration and come to the Bowman's capsule. Here, they can easily pass and come to the Bowman's capsule. And the other thing which is important is this. We have the basement membrane, and the charge of the basement membrane is negative. So positive particle can easily pass, but negative particles are repelled back again to the capillary and they cannot pass. The proteins which we have in our blood are very large, so they cannot pass through this fenestration. The red blood cells and white blood cells are larger than this fenestration, so they cannot pass. If we have any protein in our urine, if we have any white blood cells or red blood cells, it means that in the urine, it means that we have a problem in the renal corpuscle and in our kidneys. These materials, these cells or these proteins can pass through this filtration membrane and they can come to the urine. So we need to find the problem which we have, and I will talk about them later for you in the physiology. Here you can see the membrane which we call it filtration membrane. The name is here, details of filtration membrane. This is the fenestrated capillary. After that, you can see many pores here. After that, you have this blue layer, which is the basement membrane or basal lamina, which prevents filtration of large proteins. And after that, you have your podocyte. This is the podocyte with many feed process. When these podocytes feed process go interdigitately to each other, we can see some slits between them. These slits are red in their color, and through these slits, some particles which their sizes let them can easily pass, and the particles which are bigger cannot pass, and they cannot come to the Bowman's capsule, and they cannot be a part of our urine. Okay, I talked about these slides before for you, and in this picture you can see the histology of renal tubule and the collecting duct. The first tubule which we have is the proximal convoluted tubule, and this is the histology of proximal convoluted tubule. You can see the simple cuboidal epithelium, and our cell contains many microvilli. These microvilli can make the brush border for our cells. And when you have microvilli, it means that you can do reabsorption. The surface area is increased, so easily you can reabsorb more particles. And these cells contain many mitochondria because they are very active for reabsorption, and reabsorption of many particles needs ATP. The next part, loop of Henle. The descending limb of loop of Henle and the thin portion of ascending limb, as you see here, are covered by simple squamous epithelium, and reabsorption of water take place in this part. After that, we have thick part of ascending limb, which becomes simple cuboidal to low columnar cells. Amount of microvilli on the apical border of our cell diminish, but they are still active for doing reabsorption. Then we have distal convoluted tubule and our collecting duct. The distal convoluted tubules are simple cuboidal to low columnar have a simple cuboidal low columnar epithelium, and they are important for reabsorption. And this is the collecting duct. In the collecting duct, we have two types of cells. Some of our cells are called principal cells, like these, without microvilli, and some of our cells contain microvilli, and we call them intercalated cells. The intercalated cells are important for controlling the pH of our blood, and the principal cells are important for reabsorption of water. They have some receptors for aldosterone and ADH. When we have dehydration or when the blood pressure drops, the ADH and aldosterone are secreted. ADH is secreted from our hypothalamus, you know that, not hypothalamus, from anterior pituitary, because the making of ADH, happen in our hypothalamus, then it comes down through the infundibulum to the pituitary gland, and pituitary gland can release it at the time of need. And the aldosterone is the hormone which is secreted from our adrenal gland. I talked about both of these hormones when our blood pressure decreases, and how they can control the absorption of water from our kidney, and work on this part, which I will talk about them later. So you have two types of cells in collecting ducts. One type of cell are intercalated cell, which can control the blood pH, and one type of cell is principal cell. They have receptor for ADH and aldosterone. And in this picture, you can see the different parts of the nephron with different epithelium. First, we have the renal corpuscle with simple squamous epithelium, proximal convoluted tubule with simple cuboid epithelium and microvilli. The nephron loop that has two parts. One part is thin and one part is thick. In the thin part, we have simple squamous, and in the thick part, we have simple cuboidal epithelium. Then you can see the distal tubule with simple cuboidal epithelium, and after that we have collecting duct, simple cuboidal to low columnar epithelium. And I told you that these collecting ducts combine together and they can make papillary duct with simple cuboidal to low columnar epithelium, which open to the renal path. The next structure which is important is juxtaglomerular apparatus. The juxtaglomerular apparatus, or JG apparatus, is a structure which is very important for our blood pressure control and also for release of erythropoietin hormone. And this apparatus is innervated by autonomic nervous system and can regulate the blood pressure in our kidney. Now I want to talk about this apparatus and this structure. Before I talk about this slide, look at this one. In this slide, you see that. Let me repeat this part for you. This is the Bowman's capsule. Here you can see proximal convoluted tubule. This is the descending limb, loop of Henle, ascending part, distal straight, and distal convoluted. Look at this distal convoluted. Can you see a part of distal convoluted is adjacent to afferent arteriole? This structure can make JG apparatus for us. Now I go back to the slide. This is the convoluted tube, which is adjacent and it's in contact with the afferent artery. When these structures come near each other, the sum of distal convoluted tube and the cell afferent arteriole change. These cells, all of afferent arteriole, become hypertrophoid. They become a little larger, and we call them juxtaglomerular cell. And the cells in the wall of distal convoluted tubule can become taller, and their color are a little darker, and we call them macula densa. Macula densa and juxtaglomerular cells together can make these cells together can make JG apparatus or juxtaglomerular apparatus. These cells can release renin and erythropoietin. Do you remember what is the function of renin? Do you remember when our blood pressure drops and neuroreceptors detect hypotension? They send information to our central nervous system, and from central nervous system we have activation of our kidney to secrete renin. The renin is secreted from here. This renin can work on angiotensinogen of your liver, change it to angiotensin 1. Angiotensin 1 change to angiotensin 2 by the function of ACE or angiotensin converting enzyme, and then angiotensin 2 can work on different parts of our body and it can increase blood pressure. This renin is secreted from juxtaglomerular apparatus. And also the juxtaglomerular apparatus are important for secretion of erythropoietin, which can go to our bone marrow and increase formation of our red blood cells. This is this structure which we have in our kidney. And in the renal physiology, next time I wanted to talk about urine formation. We have three processes. One of them is glomerular filtration. I told you that this filtration takes place in the glomerulus. After that, we have tubular reabsorption. The material, the essential material for our body is reabsorbed here from these tubules to our blood vessels. And the next step is called tubular secretion. Still, we have some waste product in our blood vessels which need to be secreted in our tubules. The result of these three processes is the urine which is formed in our kidney. I want to stop